Aloha. My name is Clyde Tomorrow. I'll be talking about the Regional Biosecurity Project. Our specific topic is the diagnostic surveillance for koi herpes virus disease. The goal of this project actually is tied into a much larger overarching goal, and that is to expand and diversify the aquaculture industries here in the state, in Hawaii, and also in the region. All of these industries are threatened by various pathogens that can cause severe damage, economic loss. One overall idea of the project was to develop a capacity to be able to identify these specific pathogens in a very, very rapid turnaround time. To achieve this, Objective 1 was to establish a local laboratory that is equipped with PCR technology, and the focus of the initial work was on one particular disease, and this is the koi herpes virus, because number one, it is an OIE listed disease, and number two, it does represent a significant threat to the ornamental industry, both in Hawaii and also throughout the United States. Once we had the diagnostic laboratory, so we needed to develop a mechanism or process for conducting PCR testing for koi herpes virus. And with that, we started to conduct surveys both in farm and feral populations in the state. The objectives 5 and 6 are the classical technology transfer objectives, which will be producing technical handouts and also follow-up workshops. Objective 1 was achieved by forming a collaborative partnership with Moana Technologies and Dr. James Brock. Moana Technologies routinely conducts PCR testing of their shrimp stocks, so it was easy for them to incorporate KHV testing into their protocol. We obtained a positive gill sample containing the koi herpes virus, and that has been used to validate and develop the PCR testing following OIE protocol. For those of you who are not familiar with the polymerase chain reaction, this is a particular molecular technology that allows for specific segments of DNA to be amplified millions of times, and in order to be visualized on a gel. The specificity and also the ability to amplify small segments of DNA is one of its strengths. The PCR test that is used by Moana Technologies actually is following OIE listed protocols, and these include the 2 primer sequences that are used to specifically identify the koi herpes virus in the DNA. So the positive gill sample DNA was extracted and exposed to the 2 primer sequences and amplified, and as you can see, there is a positive band that is detected on this particular gel. The second gel on the right also shows a positive band, but this sequence you can see that is a different size. Second, so this particular sequence actually is coding for the gene for cytochrome oxidase subunit I. This is a positive control for fish DNA that is also going to be used as part of the testing process, as you will see shortly. Validation of the tests began with testing against species or fishes that where we know there is no possible way that they possess koi herpes virus. In this case, 3 individuals of tilapia gills had the DNA extracted and exposed to the koi herpes virus primer, and as you would expect, this is the only detection took place in the positive control. However, the same tissue or DNA samples were exposed to cytochrome oxidase primers, and as you can see, it is positive for fish DNA. So it is a combination of these 2 tests that is used to detect the presence or absence of koi herpes virus. As part of the validation process, the bands that we detected on the gels that we just saw, were extracted and purified and then submitted to the University of Hawaii laboratory where the DNA sequence was actually determined. What you see here is the actual sequences, nucleotide sequences for both the suspected core herpes virus and also the cytochrome oxidase subunit I bands. The nucleotide sequences that we obtained were entered into what is called a basic local alignment search tool or BLAST program. And this particular program helps compare primary biological sequence information such as amino acid sequences or nucleotide sequences, and helps us identify a specific sequence from a library or database. Anyway, the results of that search are presented in this slide, and as you can see, there is very, very high agreement that the nucleotide sequences that we obtain are consistent with the core herpes virus, and thus validating PCR tests. The nucleotide sequences that we obtained for the cytochrome oxidase subunit I was also submitted or entered into the BLAST program, and the search results are presented in this particular site. As you can see, we also obtained very high agreement that the nucleotide sequences are coding for cytochrome oxidase, and actually also identified Oreochromis mozamicus, the actual tilapia species that we used for the test. This also provides very, very high degree of confidence that the PCR test being used is a valid test. Objective 2 of the project, to hold a workshop and to obtain stakeholder input about the project, was completed on July 17th at Windward Community College. However, we actually combined both year 1 and year 2, where we'll be looking at diseases that will impact tilapia as well. As you can see, we had a very good turnout for the workshop. Unfortunately, we only had one individual that actually was interested in core herpes virus at the time. The next phase of the project was to establish proactive screening methods for core herpes virus, and to accomplish this, we worked closely with Dr. Alan Riggs, who's a state aquatic vet with the Aquaculture, Livestock and Support Services with the State Department of Ag. Dr. Riggs also is in charge of the specific pathogen-free screening for the shrimp industry, and it was logical to follow much of his protocols so that we would be consistent with all of the processes that we would need to be, because the core herpes virus is also an OIE listed pathogen. A major difference between the sampling protocol that we're using for core herpes virus is that we use a non-lethal gill sampling technique. This has actually great value because at the end of the day, the fish are still alive, and this is important because some individual koi are worth thousands of dollars. Anyway, a summary of the process is shown in this slide. Starts off with collection of fish from a particular rearing tank. Fish undergo anesthesia using 2-phenoxyethanol at 300 parts per million. We then take some vital statistics about the fish, and then we remove a small gill biopsy that is then fixed in 95% ethanol. Fish then are allowed to recover and then return to their holding facility. The gill samples that are preserved in ethanol are then submitted to the laboratory where they undergo PCR testing. The results of a test that is actually done at Kodama Koi Farm is presented in this slide. As you can see, when using the koi herpes primer, says and only the control gill sample turned up with a positive band, meaning that all of the other individuals tested were negative for core herpes virus. And proof of the pudding actually comes in the second test, when you can also see that all of the individual samples also are positive for fish DNA, so giving us a high degree of confidence that all of the samples are indeed negative for core herpes virus. Objective 4 was also completed over the course of the project, and we began to sample koi from a variety of locations, both in the wild farmed and also at hobbyist locations. And a summary here is shown in this particular site. Samples were obtained from Hilo, East-West Center, Wamanoa Stream, in Honolulu at a hobbyist location, and also Kurama Koi up in Mililani. A summary of the koi sampled in the wild is presented in this particular slide. Individuals were kind of few and far between. However, we did manage to capture 14 individuals, one at Vineyard Street Overpass, one in Kaneohe Stream, and a whole bunch in Manoa Stream. Results were all negative for KHV and also positive for the cytochrome oxidase, which confirms that fish were negative for KHV. We also sampled commercial vendors for koi herpes virus, and a summary of those locations are provided in this slide. Kolau Pets, Martin Pets, Waimanalo Feeds, and by far the largest sampling was done at Kodama Koi Farm. And as with the wild feral koi, all fish tested were negative for koi herpes virus and positive for the fish DNA. We also sampled koi that were at displays or owned by hobbyists throughout the state. East-West Center in Waimanalo, Pat Maloney's, Aquaculture Farms in Hilo, this is Howard Takata's stock, and also Glendang in Honolulu. And as with all other samplings, KHV was not detected in any of the samples. It has been reported in the literature that KHV DNA has been detected in other cyprinids, such as in goldfish, and in goldfish in particular. We took the opportunity to sample comets and goldfish at Kolau Pets, and as is the case with all of the samples to date, all were negative for KHV. So far, we have no indication of any koi herpes virus individuals here in the state of Hawaii. And so what does this particular result mean? To answer that question, so we have to take a look at the industry itself, and the ornamental industry currently accounts for about 10% of the aquaculture output in the state of Hawaii based upon 2008 statistics. We wanted to look at the ornamental industry closer, and we looked at there is actually consists of both marine and also freshwater ornamental industry components. Based upon work done by Cam et al. Published in 2005, we can see that the koi represent by far the highest percentage as far as the farm gate value output in the state. More recent data regarding the freshwater ornamental industry in the state shows the impact made by one particular farm, and this is Kurama Koi. Anyway, from 2006 and before, the ornamental industry in total, both marine and freshwater, were about $300,000 to $500,000 as far as gross revenues. However, after 2006 and when Kurama Koi came online, you can see a significant rise in the outputs, and this is due primarily because of this one farm. So roughly over $2 million of farm gate revenues being generated by this one farm. This presents a very, very good opportunity to utilize the results of this project, as the opportunity has arisen which we may be able to achieve specific pathogen-free status for Kurama Koi, and by doing that, perhaps we'll be providing some added value to his outputs. It may not be as critical for the United States, however, because he markets both within the US and also internationally, this particular certification may have significant value, especially overseas, and forms the basis for future work.